I'm Laurie Hutcheson, Executive Director of the Lobular Breast Cancer Alliance, and I am happy to welcome you to this conversation with our new AACR-LBCA Deborah Mueller Foundation Fund Invasive Lobular Carcinoma Innovation and Discovery Grant Grantees, quite a mouthful. Welcome. Today, we're going to be hearing from Doctors Carolina Reduzzi and Julia Houthuijzen, and the patient advocates they worked with, Julia Levine and Siobhan Freeney. And we are also very happy to welcome Dr. John Doski to this conversation. As you know, this grant is named in memory of John's wife and our wonderful board member, Dr. Deborah Mueller, who passed away precipitously from metastatic lobular breast cancer 2 years ago. Deb was an ardent supporter of lobular breast cancer advocacy and research, and gave us her all during her short time that she worked with us on the board. We truly miss her. And we are so grateful that John and his daughters, Julia and Sarah, have made these grants possible and have been so committed to sustaining Deb's legacy and ensuring that important lobular breast cancer research continues to happen. Welcome to all of you, and congratulations to our grantees. The format for today is that we'll first hear from the grantees about their research and the patient advocates with whom they worked. And then I will kick off with questions, and everyone else can chime in as well. I'm going to ask each of you to introduce yourselves and tell us where you are geographically, because we're all over the place in many time zones. All righty. So now I'd like to ask Julia and Carolina one at a time to share their research with us. Please tell us your study title and all about your proposal, what you hope to demonstrate, the data that supported your work, and how you have and will continue to work with your patient advocate. And then I'm going to ask each of the patient advocates to say a bit about what it has been like working with you as the grantee and on this study. And then we will open it up for more questions, and I'll invite everybody to jump in with questions as well at that time. So I think we're now going to start and hear from Julia, and I'm going to go ahead and share your screen if you have some slides that you'd like to show us. Julia?
Yes, thank you so much for giving us the opportunity to share about our research. My name is Julia Houthuijzen. I am I'm a senior postdoc in the Netherlands Cancer Institute in Amsterdam. And I would like to tell you a bit about the research proposal that I submitted for this grant and the progress that we have made so far. So a little bit about myself. I have obtained my PhD about 8 years ago, working already on the interaction between the tumor and the microenvironment. And this has really sparked my interest in investigating how tumor cells interact with their normal cells that are surrounding them, and how this affects tumor progression and therapy response. So hopefully be able to share my next slide. So our focus is on invasive lobular breast cancer, and this breast cancer subtype, it's the second most common type of breast cancer amongst women, and it is characterized by a very distinct growth pattern, and it has it's very different from other breast cancer subtypes. So what you can see here on this image, this H&E image here, is that you see these rows of tumor cells that are interleaved with a lot of stroma. And this stroma is rich in fibroblasts, which are stained here at the bottom in brown. And when you look at this morphology, what it kind of reminds you of is this sort of layer cake that we get, where the cake is the tumor cells and the frosting in the middle is the stroma that contains a lot of these fibroblasts. These fibroblasts are called cancer-associated fibroblasts, and they play a very important role in helping tumor cells grow, metastasize, and also become resistant to therapy. And we are interested in looking at this crosstalk between the tumor cells and their microenvironment, as we call it, because there is a lot of communication between these two. So there's constant communication between the tumor cells and their environment, and there's actually molecular cues being sent back and forth to these cells. And as you can imagine, looking at this H&E image again, you can see that there's close interaction with basically all of the tumor cells and their surrounding stroma. And this crosstalk plays a very important role in tumor growth, metastasis, immune suppression, and ultimately also therapy response. However, when we study ILC, we run into a couple of problems regarding the model systems that are available for this research. You can imagine that in the lab, we like to use certain model systems to study the disease in more detail and come up with novel therapies or strategies to manage disease. For that, we use either cell lines in culture, mouse models, or PDX-derived models where we take samples from patients and inject them into mice. However, if we take a look at the amount of models that are available to study ILC, this is very limited. For instance, the ILC cell lines that are available, there are less than 10 generated over decades of research. Whereas to compare this to another breast cancer subtype, which is triple-negative breast cancer, which is a very aggressive subtype, there we have over 50 cell lines available to do our research, despite the fact that this subtype of breast cancer is only accounts for 5% of all breast cancers, compared to ILC being represented by 15% of all breast cancer cases. And moreover, the majority of these models do not or only partially maintain the tumor microenvironment that is so important for therapy response and the way that these tumors grow and metastasize. This means that there is a need for a model system that closely recapitulates the unique ILC architecture and the crosstalk between the tumor and the stroma in order to study this disease in a reliable way. In order to set up such a platform, we have made use of our genetically engineered mouse models that we have in the lab of Jos Jonkers here at the Netherlands Cancer Institute, where we can model ILC in these mice. By genetically altering the genes that are also frequently found to be mutated in human ILC, these mice develop tumors that look a lot like human ILC in terms of their growth pattern and also their tumor microenvironments. We can harvest these tumors from these mice and we can section them into 250 micrometer thin sections that we can keep alive short-term in culture. And we have analyzed these using different methods. So we've looked at the cellular composition, both by flow cytometry and histology, and also looked at the RNA expression once we took these tumors into culture. What we found is that for short terms of culture, so up to 7 days, we can keep these tumor sections alive and happy. The morphology closely mimics what we see in vivo, and also the tumor composition remains quite diverse. So we maintain our cancer-associated fibroblasts, shown in blue, our tumor cells. We do see a reduction in the amount of immune cells in these tumors, but up to 3 days, this is still very comparable to the in vivo situation. Moreover, what we also noticed in these ex vivo cultures is that the therapy response that we see in our mice is very closely mimicked in our ex vivo cultures. So, for instance, in our mice, we see a good response to PI3 kinase inhibitors, which is called here the BES235. However, we do not see any response to immunotherapy, which is also known from literature. Standard chemotherapies like doxorubicin have somewhat of an effect on the tumor growth, but not massively. And this dynamic range that we see with different therapies creating different responses in these mice is also something that we can mimic in our ex vivo platform. So again, here we see that tumor cell proliferation is reduced when we administer doxorubicin to these ex vivo cultures, but is even further reduced with the PI3 kinase inhibitor. Moreover, also here, the immunotherapy does not result in any increased cell death in our ex vivo cultures, again, mimicking what we see in our mouse model. So our next step was, of course, to take this platform away from the mouse models, but also try and see if we could incorporate this with human ILC samples. And that's where this grant came in, and I applied for this funding, and found luckily that I received the grant, and it really fit with what we want to achieve. So what we are doing currently is to isolate ILC samples from patients that go in for mastectomy or local removal of the tumor here at the Antoni van Leeuwenhoek Hospital. We can obtain these tissues and perform the same type of experiments as we have done with our mouse models. So again, we take these tumors, we slice them into thin sections, and we can culture them short-term and analyze them in different ways. And the overall aim of our study is to establish this ex vivo culture platform also for human ILC, to find ways to cryopreserve these specimens so that we can also use them for later use. And also to, of course, utilize this platform to perform sort of high throughput drug screening of both existing therapies, but also novel cancer therapies. And in that way, we hope that these tumor slices can function as avatars that we can, that mimic the response of a therapy in patients. We have some preliminary data from a first ILC patient that we received tissue of. This was a relatively young patient in her early 40s, and we were able to obtain these tissues after she underwent a mastectomy. And we were able to keep these tissues alive quite happily up to 7 days. Also, the morphology is maintained of these tumors in culture. And moreover, we were able to perform a drug screen where we combined endocrine therapy together with a CDK4/6 inhibitor to see differential responses to therapy. And what you can see here in this graph is increasing doses of a drug that we gave to these tumor slices, where we see a clear response to tamoxifen, so this is the endocrine therapy, and also a response to the CDK4/6 inhibitor. But upon combination, we see an even better response. And this is the first data that we have obtained within this grant, and hopefully we hope to include many more patients and be able to build a sort of biobank where we can look at therapy response in patients and be able to stratify patients based on either their sensitivity or resistance to drugs. And of course, also any interesting findings that come from our other lines of research in the lab where we try to target these, specifically these fibroblasts, can also be tested immediately in this platform. And with that, we hope to push the research of ILC, also in a human setting, forward using relevant clinical models to study this disease in more detail. And with that, I would like to thank the people that are involved. So my team is slowly expanding and contains of Constantina, who's over here in the picture. She's the one that pioneered this platform and started working towards also incorporating this in for the human setting. We have Daisy, who is her student that helps along with this, Pim, our bioinformatician, and the other people involved. And of course, I also want to thank Siobhan for being the patient advocate on this project and providing valuable feedback for this. And of course, all the other people that are involved in the practicalities of doing research. And of course, our funding organizations for allowing us to do this wonderful work. Thank you.
It's very exciting to see. So I think before I ask any questions, Siobhan, I'd love to hear, first of all, where are you? And tell us a little bit about yourself and then your experience on this grant so far.
I'm sitting in my house, which is on the southeast coast of Ireland, and it's afternoon here, so really happy to see all of you. I know some of you. I I suppose just to set an understanding for people, I got involved with lobular breast cancer research when I was invited as one of the first patient advocates to a meeting of the ELBCC, the European Lobular Breast Cancer Consortium, and that was about 5 years ago. And I've been passionately and very active in that. So look, when you contacted me, I was just immediately taken with the whole idea of, for me, the importance of mouse models. I had visited another lab not too long ago and visited the mice and have a reasonable understanding of that. And it's just so important to get these mice models made available for research right across the board. But also the added importance of working on human ILC patient samples is just going to be a game changer. And we're seeing it, it's happening. But yours is exciting, it's new, you've got results already. And I believe that this will change therapeutic actions and much needed patient care and outcomes.
We try our best, of course, to keep in mind as many of the aspects of the tumor as possible, but still keep it like a practical and workable model system that we can handle in the lab. And I think that's really been sort of the main, a point of view for us to take off from, to be able to study this tumor microenvironment that is so important in directing therapy response and the way that these tumors metastasize. That it is, you cannot properly do research without when you ignore these kind of factors, I think. And we try to recapitulate that as good as possible.
And of course, you mentioned biobanking as well. And
yes,
that is something that's so badly needed. And it's exciting to think that this will lead to, the beginnings of a biobank, which again, then, will build on everything that you've done, by the end of the project.
We're currently, pioneering the cryopreservation with our mouse tissues, because the human tissues that we receive are so precious that we want to use them mainly for the drug screens that I mentioned. And also looking at, ways to study invasion. So we're pioneering that with the mouse tissues to see if we can cryopreserve it, because that would indeed be a very valuable asset if we can biobank these slices, that even, if a patient comes back with a relapse, we can go back to tissues that are in the freezer and we can test new types of drugs if we want to. And also with all of the treatments, the progress that's being made over the last couple of years with all different kinds of therapies, that we can go back to these tissues also later on in like 3 or 5 years time and test new hypotheses and new ideas to push the research forward.
Yeah, no, so important that we will have that, so that it's not just a one time, that, as you say, you can go back and you can check and you can test them with different targeted therapies and treatments.
Yes, and that's also one of the benefits, because of course a lot of people put also efforts into generating organoids, which I think is a very interesting avenue to explore as well. But again, you lose the TME composition. The added benefit of having organoids, you can keep them alive for much longer than that we can at this point in time. But it takes time to establish these cultures and be able to do the drug screens that you as a scientist envision. And most of the times patients don't have this months of time to wait for what comes out of the lab, right? And then having a sort of on the spot kind of test where you can cut these tissues and within a week you will know what the response is, I think is going to be very important.
Yeah, and organoids are hugely important, but just, the fact that it's live, mice, and they mimic the mammary glands, and so it's way more important that we have these. Sorry,
Julia, what is this platform that has been created something that will be replicable and shared, like open access with other researchers?
Yes, we are actually in the process of drafting a manuscript for the platform that we've established with the mouse tissues. And we hope also to include some first sneak previews of the work we are now currently doing with the human ILC specimens. So that manuscript is in preparation, and we hope to submit that end of this year. So it'll definitely, all of the details will be made available to other researchers, of course. And we're also actively collaborate with other people within the Institute and also outside of the Institute that are interested in using this platform also for other tumor types.
Great. I think we can have more questions after we hear from our next grantee. So I want to give Carolina a chance to go ahead.
Perfect. So I'm Carolina Reduzzi. I'm a PhD and assistant professor of cancer biology research in medicine at Weill Cornell in New York. I'm very excited to be here and present today this proposal that was awarded and the title of the proposal is Compressive Liquid Biopsies to Decipher Lobular Breast Cancer Metastasis. And this proposal was prepared together with Julia Levine, our patient advocate. She will talk more later about how we work together on this, but I just wanted to acknowledge her right away because among the things that she did during the preparation of the proposal was to give some feedback about the title. And the final title that we chose was one of the ones suggested by her, and I really liked it. So thank you, Julia, for this. So a little bit of background on ILC. I don't think there is the need in this context, but we know that it's the second most common histology of breast cancer, and it is a different entity from invasive ductal carcinoma, but regardless, these two subtypes are still clinically measured as a single disease. Among the differences of ILC, there is the fact that it has a specific metastatic spread, leading to organ dysfunction and quick clinical deterioration, together also with the late recurrence, as we were mentioning before. And therefore, there is an unmet need to better characterize ILC and its peculiar metastatic behavior and implement precision medicine-based approaches for patients with ILC. So in this context, liquid biopsy is a precious tool to advance a personalized approach. Liquid biopsy means to analyze various tumor-derived material that can be found in the blood of patients with breast cancer. Among these tumor-derived material, there are circulating tumor cells clusters that are groups of circulating tumor cells traveling together in the blood, and cell-free DNA. Liquid biopsy can be used for a number of applications, but the most commonly used are for cell-free DNA, the molecular profiling of the tumor in real time to both detect possible therapeutic targets and also monitor the response to treatment, while CTCs have a proven prognostic impact in breast cancer. And because they are cells, they can be also analyzed to study the biology of the tumor, such as the metastatic process. And in particular, CTC clusters, because they are clusters of cells, they have been demonstrated in vitro and in vivo they are considered to be the true seed of metastasis because they have enhanced survival in circulation, and then when they arrive to secondary sites, they are not single cells, but they are groups of cells, so they have a higher metastatic potential. So in this context, including liquid biopsy, including both circulating tumor DNA and circulating tumor cells, appear as a useful tool. But to date, liquid biopsy studies that are extensive in breast cancer in most cases have not been focused specifically on lobular breast cancer. So with our group, we have done some studies about liquid biopsy in breast cancer and also in lobular breast cancer. And this is a multi-institutional analysis done on ctDNA data from patients with lobular and ductal breast cancer, metastatic, where we compare them to see the genomic landscape of these two tumors. And we confirm something that was already known, that a mutation in CDH1 that are particularly frequent in lobular breast cancer. And we know that these alterations lead to the loss of E-cadherin, which is the hallmark of ILC. In an independent analysis on circulating tumor cells, we also saw that alteration in CDH1 are associated with a higher number of CTCs and a higher number of CTC clusters. This was not done in ILC specifically. So the fact that alteration in CDH1 are associated with a higher number of CTCs makes a lot of sense, because we know that loss of E-cadherin in vitro gives an enhanced migration and invasion capability to the tumor. However, it's a little paradoxical when it comes to clusters, because for the evidence that we have so far about clusters, they are characterized by a high adhesion between tumor cells. At the same time, most of the studies done on clusters have not been focused on lobular breast cancer, but on ductal breast cancer. So it might be that in lobular breast cancer, these clusters are associated to CDH1 alterations and to loss of E-cadherin, but that would mean that maybe they are different from the clusters that we see in ductal breast cancer. Another thing that we observed for CTCs in lobular versus ductal breast cancer is that they have a different prognostic significance. So if we see here on the left, it's the analysis of the prognostic impact of CTCs in ductal breast cancer, and we see that we can clearly distinguish patients with better and worse overall survival based on a established cutoff of 5 circulating tumor cells per sample. But when we do this analysis in lobular breast cancer, specifically on the right, we see that there is no difference in survival when we use the cutoff of 5. Also no difference when we use the cutoff of 80 CTCs, while we see a prognostic impact using a cutoff of 100 CTCs. This, again, is probably due to the fact that, as we saw, lobular breast cancer release many more CTCs, but at the same time confirms that also from a liquid biopsy perspective, ILC is different from IDC. And so the analysis of CTC and CTC clusters in this subtype specifically might be a useful tool to understand the biology of ILC and how it is different from IDC. So based on this preliminary data, we hypothesize that CDH1 alterations in ILC contribute to a higher release of both CTCs and CTC clusters in the circulation, which determine the peculiar metastatic behavior of ILC compared to IDC. And we think that by understanding this relation through liquid biopsy analysis, we might decipher the mechanisms underlying ILC metastatic spread and potentially identify novel treatment targets specific for this type of breast cancer. Because in the context of CTC clusters analysis, since they are considered the main seed of metastasis, there is a research trying to understand what are the target of cluster formation, to use that as a potential treatment to prevent metastasis formation. But if the mechanism that underlie clusters in IDC and ILC are different, then also the strategies to try to stop this cluster formation might be different. So we defined 2 specific aims. The first was to evaluate the association between circulating tumor DNA alteration and specifically CDH1 alteration with the presence of CTC clusters in lobular metastatic breast cancer, because we have some indication that there is this association, but nobody really did it in a specific cohort. And also to explore differences in gene expression between single and clustered CTCs to unveil the mechanisms responsible for cluster information and possibly identify novel biomarkers in lobular breast cancer. So for the first aim, this will be a retrospective analysis in which 30 patients with metastatic ILC will be compared to 30 patients with metastatic IDC that will be matched for treatment line and subtype. These patients will be analyzed for ctDNA alteration with the Guardant360 assay and for CTC with the cell search platform. And then we will evaluate if there is an association between the presence and size of clusters and CDH1 alterations. At the same time, 20 patients of these 30, 20 patients will process in parallel with the different technologies for CTC enrichment, which is based on is not based on epithelial markers for CTC enrichment. And that, because we don't have a lot of time, I didn't go too much into these details, but basically cell search, in order to enrich and identify CTC and CTC clusters, uses the fact that these cells express epithelial markers. But at the same time, we know that ILC have a decreased expression of EpCAM, for example, that is a key epithelial marker for the cell search function. And so we want to see if by using a platform that is based on physical properties to enrich and detect CTCs might increase the detection of clusters. For the second aim, we will perform a prospective analysis. So the 20 samples of the aim 1 that will be prospectively collected and analyzed with Parsortix will also undergo isolation of CTCs and CTC clusters using a micromanipulator. This will be done both in 20 ILC patients and in 10 IDC patients, also matched for treatment line and subtype. And then the isolated CTCs and CTC cluster will undergo RNA analysis. And this will allow us to compare transcriptomic profiles of CTCs and clusters in ILC versus IDC. Of course, this is a small cohort because it's a one-year grant, and we hope that this is just the starting point of a bigger study where we can include more patients and really try to define what are the transcriptomic features associated with clusters in ILC versus IDC. And here I wanted to, so this was what we presented and we submitted, but in the meantime, we did this additional analysis that I think is interesting. We submitted it for the San Antonio Breast Cancer Symposium, in which we tried to see if the characteristic of the clusters in IDC and ILC patients were different. And we actually saw that, yeah, there are more clusters in ILC, but they are smaller in size, so less, each cluster contains less tumor cells. And also they have a higher frequency of white blood cells attached to the clusters, because CTC cluster can be homotypic, meaning including only tumor cells, or can be heterotypic, so including both tumor cells and white blood cells. So also, it's not only a difference in terms of number, but also in terms of quality of these clusters, suggesting even more that these clusters are different, and hopefully we will be able to really understand what is the difference between these clusters. And that's the end of my presentation. I want to thank my lab, the Cristofanilli CTC lab, Massimo Cristofanilli, who is my mentor, Julia, and of course the funding agency that funded this grant. And thank you for listening to me today.
Thank you so much, Carolina, and for trying to condense that into so few minutes. before I ask a couple of my basic questions, I want Julia L. to have a chance to talk about her experience with you. And it sounds like she was really influential right from the beginning. So, Julia, you want to tell us about your experience and the study? And unmute yourself, please.
Good morning. Excuse my voice, I'm a little hoarse. I am sitting in Venice, California. It's 7 a.m., so it's a little dark in the background. I was first contacted by Carolina by email. I'm not actually sure how she got my name, but I had already been familiar with Dr. Krista Finelli from the very beginning. I should say that I was a founding member of LBCA starting way back in 2016 from the first International Lobular Symposium. And after that, I became very, very involved with all the research, including putting together a publications library on the LBCA website. And, firsthand noticing obviously the lack of ILC research and the lack of ILC in clinical trials. I should also say I am a de novo metastatic patient. So anything that involves metastatic ILC that is being studied, which is very few, is catches my eye. So as soon as Carolina contacted me, it was kind of a no-brainer. I am interested in liquid biopsies. I had already read the paper she showed by Davis, who was the first author, from her initial study, the precursor. So I was familiar with that paper a little bit, comparing IDC and ILC, and that was very interesting. Additionally, I was a very early user of a CTC blood test. I think it was 2019 or 2020 when I had initially heard about it for myself. So when she sent it, what was great about it was, there wasn't that much time. I think there was maybe a couple of weeks, or it was right at the same time as AACR. So I know that she was slammed and everything was rushed, but I had really an opportunity to comment on all the proposals, the scientific proposal, the lay summary, and whatever else has to be submitted. So there were, I think, 3 different documents that we worked back and forth on. And I think that went really well. And it was a great experience, and it wasn't too last minute. And I'm just excited that there's more studies in metastatic ILC and in the liquid biopsy arena, because I think it's kind of the next wave of what's going to help metastatic patients. I also just want to acknowledge Deb and John. I met Deb. I got to know her in San Antonio, I guess 2 years ago or so. Both Deb and John welcomed us advocates and scientists into their home, and he was gracious enough to offer it again after Deb passed away. And her death was a shock to many of us, and I'm honored to be a part of a grant in her memory, and I want to thank John. And this is why we do what we do. Thank you.
Thanks so much, Julia. I here. I agree with everything. It's a study, Carolina, that for me it's so needed, because we've been hearing about the promise of liquid biopsies for predicting things and for, assessing, as you're talking about, the potential or the likelihood for metastasis, and so little has been done focusing on lobular breast cancer, a metastatic lobular breast cancer in particular, and it's just so needed. So I'm really excited, and I guess my questions are a little bit, if you could just explain the retrospective analysis and the prospective analysis, just very simply, the two aims. Again, is it in both cases to compare the IDC CTC clusters in IDC versus ILC, or can you say more in how they different, and also what is Guardant 360 and cell search?
Oh, sure. Sorry. So Guardant 360 is the most commonly used clinical test for ctDNA alteration in solid tumors overall, not just breast cancer, is used in the clinic. And it's a targeted sequencing panel that investigates the presence of somatic mutations or tumor mutations in a set of commonly mutated genes in cancer. While cell search, on the other hand, is the only platform that is currently approved for use in the clinic for CTC enumeration in breast cancer. So for these two tools will be used in the retrospective analysis. So the retrospective analysis is only based on clinical testing, which is why we separated it, because it can be, we are hoping to better understand how these two biomarkers, the CTC and CTC cluster and the ctDNA, work in ILC versus IDC with some tools that can be directly used in the clinic and are currently used in the clinic, but based on evidence that derived from mainly IDC. So for example, the 5 CTC cutoff that is used to stratify the patients for prognosis probably is not the right one to be used in ILC. And in particular, in that analysis, what we want to do is to really demonstrate if there is an association between the presence of clusters and CDH1 alteration in ILC specifically, because we saw that there is an association between CDH1 alteration and the presence of clusters, but that was done in an overall cohort. So we want to know, is that something that only in ILC cause the release of clusters, or that's something that happens overall, but simply CDH1 alterations are not that common in IDC. But whenever you have a CDH1 alteration, you will have more clusters. So that's what we want to understand with that aim. While in the second aim, what we want to do is to characterize the clusters. So regardless of the CDH1 alteration, the clusters that we see in IDC versus ILC, are they similar? Are they deriving from similar pathways, similar mechanisms, or not? And based on that, can we target them with the same strategies or not?
Thank you.
Can I just add?
And sorry, I was going to say, Julia, and everybody, I'm just going to open it up to anyone's questions at this point, and we'll go, for as long as there are questions. And so go ahead.
Can I just add something to what Carolina said in terms of your question? So Guardant 360 is one of several companies that do genomic testing on your blood, and for looking for mutations that will help in treatment decisions. And they're used usually upon progression. Some people are using them serially, but they usually will show, they may or may not show CDH1. So, but that's being used in general for metastatic patients. When you hear about genomic testing, that's what she's talking about, usually what people are talking about, or biomarker testing. CTC, which is cell search, is not widely used, and is, I guess, is not that new a technology, right? But it's just not as widely used. So hopefully this study will, help, add another thing to the toolbox for metastatic patients. Does that help explain it too?
Yeah, thank you. Thank you, Julia. Yes, of course. Everyone is invited to join us around the world on October 15th. We're declaring it Global Lobular Breast Cancer Awareness Day. And there are events happening now that we planned around the US at different hospitals, webinars, presentations, and press conferences, and also everywhere around the world. And if you go to our website, you can go to the October 15th webpage, and you can see, are there events that are happening virtually, where they are, what they are, and whether you can participate if you're not in the immediate vicinity. And pretty much every week, new places are being added to the map, both within the US and outside of the US. And I have been in touch with members of Congress to talk about declaring October 15th as a National Lobular Breast Cancer Awareness Day. So if not this year, it'll be next year. But we're anyway hard at work on that. Thank you for promoting it, and we invite everybody to check it out and join us in individual acts of advocacy, or join one of the group events happening. We are really, really shouting out now about lobular breast cancer and getting more attention and more research. All right, well, I want to thank everybody for joining this great conversation. The presentations have made me so hopeful about the advancement of understanding of lobular breast cancer, metastatic lobular breast cancer, the prospect that we may soon have the identification of lobular specific treatments. Brava. I look forward to hearing from you both at the end of the grant period, if not before. And I hope to see you in Europe, or at San Antonio or ASCO, where we hope to have a lobular presentation as well in the spring. All right. I think that concludes this conversation. Thank you all.